Every single living thing, from the tiniest microbe to you and me, depends on this one ancient, incredibly precise process. It's life's ultimate copy machine. It's DNA replication. So let's pull back the curtain and see exactly how life's blueprint copies itself, billions of times, all across the globe, every single second.
I mean, just think about it for a second. You started out as just one single cell, and now you're made of trillions. Every one of those new cells needed a perfect, exact copy of your genetic instruction manual. So how does nature pull off this incredible feat with almost perfect accuracy, over and over and over again? Well, that's the huge puzzle we're going to piece together today.
All right, so before we can jump onto the factory floor, we kind of need to understand the blueprint itself. And this knowledge? Well, it didn't come from a single eureka moment. Nope, it was pieced together over the course of nearly 100 years.
Okay, so the story really kicks off way back in 1869 with the discovery of this stuff called nuclein. But then it took almost another century for Watson, Crick, and the incredible Rosalind Franklin to figure out its iconic double helix shape. But the real juicy secret, how this thing actually copies itself, that wasn't unlocked until 1958 thanks to a really clever experiment. And what Meselson and Stahl proved was just, well, it was beautiful in its simplicity. They showed that replication is semi-conservative. And all that means is that each new DNA molecule is a hybrid. It keeps one of the original, old strands, and then it builds one brand spanking new strand right alongside it. It's a brilliant model that ensures you get a perfect, stable copy every single time.
Okay, so with that history lesson out of the way, let's go inside the cell. We're heading to the factory floor. We're about to watch a microscopic assembly line that runs with a speed and a precision that is just mind-boggling. And it's all thanks to a specialized crew of molecular machines.
So, this whole assembly line, it basically runs in three main phases. First up, you've got initiation. That's all about getting things started. Then comes the main event, elongation, which is where the new DNA is actually built. And finally, there's termination, where everything gets cleaned up, double-checked, and sealed. So, let's break them down. Right, you can't copy a book if it's slammed shut, and you definitely can't copy DNA when it's all coiled up. So, the initiation phase is all about finding the exact right place to start. It's called the origin of replication, and then carefully unzipping the two strands of that double helix to get at the code inside.
And to do this, you've got this incredible crew of enzymes. Leading the charge is helicase. It literally spins like a helicopter blade to unwind the DNA. But all that unwinding creates tension down the line, right? So topoisomerase runs ahead of it to relieve that stress and prevent tangles. And to make sure the two strands don't just snap shut again, these little single-strand binding proteins act like tiny paper clips, holding the blueprint wide open.
Okay, so the blueprint is wide open. Now the main event, the main construction phase, elongation, can finally begin. This is where the star player of our factory floor gets to work, reading the old template and building the new strands. And the star of the show here is an enzyme called DNA polymerase. This thing is the master builder, the photocopy machine of the cell. It expertly adds the new letters of the code, one by one. But here's the catch, and it's a weird one. It's a master builder that, for some reason, can't lay the very first brick. And that is where our next little enzyme, primase, swoops in to save the day. Primase lays down a short little strip of RNA, and we call that a primer. This primer is the crucial go signal, the starting block that DNA polymerase needs to grab onto and start building. Okay, now this is where things get really, really clever, because the two DNA strands are basically mirror images of each other. They run in opposite directions. They have to be copied differently. One strand, which we call the leading strand, gets built in one smooth, continuous go. The polymerase just follows the helicase, just cruising down a highway. But the other strand, the lagging strand, has to be built backwards, away from the action, in a bunch of short, disconnected chunks. So these little disconnected pieces on the lagging strand, they actually have a name, Okazaki fragments. And each one needs its own little RNA primer to get started. So the lagging strand is built in this sort of back and forth stitch pattern, a little piece here, then another piece there. It's definitely more complicated, but it gets the job done perfectly. So we've got two new strands of DNA, but hold on, the job's not done yet. We've still got all those little temporary RNA primers to get rid of, and all those Okazaki fragments on the lagging strand need to be joined together. This is the cleanup phase, termination. This is where a special cleanup crew comes in. First, an enzyme zips through and snips out all the temporary RNA primers. Then, our friend DNA polymerase comes back to fill in those empty spots with the proper DNA. And then, the real hero of this final phase, DNA ligase, acts like molecular glue, sealing up every last nick and gap to create one continuous, perfect strand. But here's the most mind-blowing part. How is this whole thing so accurate? Well, that master builder, DNA polymerase, is also a master proofreader. Seriously, think of it like a typist who has a backspace key. It checks its own work as it goes, and if it accidentally puts in the wrong letter, it immediately stops, backs up, and fixes it. So with all this quality control, what's the error rate? We're talking about one mistake in a billion letters. I mean, that is just an astonishing level of precision.
Whew, that was a lot of different players on the field, right? So let's do a quick final roll call for our all-star factory crew. So you've got your unzipper, your starter, the main builder, the stress reliever that keeps things from getting tangled, and of course, the finisher that glues it all together. It's a perfect little system where every single one of these molecular machines is absolutely essential. If even one of them fails, the whole assembly line just grinds to a halt.
Okay, so after all that unzipping, building, proofreading, and gluing, what are we left with? What's the final result?
We end up with two perfect, identical DNA molecules. And if you could look really closely, you'd see that each one is a perfect hybrid, just like Meselson and Stahl predicted. One old parental strand and one brand new daughter strand. That one single blueprint has now become two, each one ready to be the instruction manual for a brand new cell.
And that leaves us with one last really fascinating thought. This system is designed to be almost perfect, right? An error rate of 1 in a billion. But in a genome with billions of letters, that means mistakes can and absolutely do happen. So the question becomes, what happens, for better or for worse, when that 1 in a billion mistake actually slips through the cracks? Well, that, right there, is the engine of all genetic variation and evolution itself.